further catabolized into carbon dioxide and water. Whereas in broader sense, physiological or macroscopic respiration, it means the multicellular organisms, they are inhaling oxygen and releasing carbon dioxide. Okay, now here this is about the molecular processes by which cells consume oxygen. That means the oxygen we inhale, that oxygen actually goes to the cell. That is the cell is consuming oxygen, and then after the respiration there is the production of carbon dioxide. And how this happens? This happens in three stages. Okay, there are three major stages by which the cell consume oxygen and produce carbon dioxide. The very first stage is the where the organic nutrients, for example, glucose, fatty acid, and amino acids, they are oxidized to two carbon compounds, that is acetyl CoA. So here, if in the figure, if you look at this, the very first stage is the production of the two carbon compound acetyl CoA, which is actually produced from the different nutrients, whether it is glucose, fatty acid, and amino acids. Okay, so we have started the glucose, and we have learned this one, glucose to pyruvate by glycolysis. And you can see in this figure, in the next step, pyruvate will be converted into acetyl CoA, and you can see other two nutrients, amino acids and fatty acids, they are catabolized into the two carbon compound acetyl CoA. And when this catabolism occurring, electrons are released. So you can see that electrons are released from each of the nutrients. Okay, and then the acetyl CoA, which is produced after the catabolism of the nutrients, those acetyl CoA enter the citric acid cycle. Okay, so this is like the second stage where the acetyl CoA is oxidized by the citric acid cycle. And here we can see during the oxidation of acetyl CoA, electrons are released, as well as we also can see the production of carbon dioxide. All the electrons which are released during the stage 1 and as well as stage 2, they are accepted by the electron acceptor or the coenzyme NADH and FADH2, because they are the electron carrier. So their function is to carry the electron to the electron transport chain, also known as respiratory chain, where these electrons are used to reduce the molecular oxygen into water. At the same time, ATP is synthesized from ADP and inorganic phosphate. This is also known as oxidative phosphorylation, because ADP is phosphorylated into ATP, that is oxidative phosphorylation. So if I ask you, at the stage 1, where we can see the production of acetyl CoA, what kind of metabolic pathway you will see here? From the different nutrients we get acetyl CoA.
Glycolysis.
Converging catabolism.
Yes, this is converging catabolism. And the second one?
Diverging anabolism.
Second one, stage 2. Cyclic pathway. This is cyclic pathway. মানে কি ছিল? Converging catabolism, diverging anabolism, and cyclic pathway. So this is cyclic pathway, the second one, acetyl CoA oxidation. আমরা এখনো diverging anabolism এ যাইনি, because this is not about anabolism. We are still in the catabolic phase. Clear?
Yes, ma'am.
Okay. So we are going to learn the citric acid cycle, which is also known as tricarboxylic acid cycle or TCA cycle or Krebs cycle according to the name of the discoverer, Hans Krebs. And in this cycle, the carbon skeleton of actually everything which are coming from here, again if you look at this, acetyl CoA is actually coming from glucose, fatty acids, amino acids, and they are then going to the citric acid cycle, where it is further oxidized into carbon dioxide and electrons are released. Okay, so because we are still in the metabolism of glucose, we have learned up to the production of pyruvate from glucose. We have to learn the next step where pyruvate is converted to acetyl CoA before the acetyl CoA enters the TCA cycle. So pyruvate is actually oxidized, this is also oxidation reaction, oxidized into acetyl CoA and carbon dioxide. Okay, so we get two products from pyruvate by this reaction. One is acetyl CoA and carbon dioxide, and the enzyme which catalyzes this reaction is called pyruvate dehydrogenase complex or PDH complex, and this is a cluster of enzymes. So here in this reaction, not only one enzyme is functioning, there are cluster of enzymes, several enzymes are involved here, and this reaction is actually called oxidative decarboxylation. So oxidation is happening, at the same time you can see carbon dioxide is produced, so that is the decarboxylation, oxidative decarboxylation reaction, and this is irreversible oxidation. So this is not reversible reaction, it will go only one way, where the carboxylic group that is removed in the reaction, you can see the pyruvate, the carboxylic group is marked with pink color, that is released as carbon dioxide, and the two remaining carbons become the acetyl group of acetyl CoA. Okay, and you can see in this reaction pyruvate dehydrogenase complex, which is actually made up of three enzymes, E1, E2, E3, and these enzymes they use several coenzymes or prosthetic groups. So here are they: coenzyme A, NAD plus, TPP, lipoid FAD. And another important outcome, the generation of one molecule of NADH. So this NADH actually carrying the electrons which are released during this oxidation, and that electrons will be ultimately delivered to the electron transport chain. Okay, now that is not the end of this reaction. There is more to this reaction. This enzyme, pyruvate dehydrogenase complex, this is a cluster of enzymes and it has three enzymes, and multiple copies of these three enzymes are present in this cluster, and they are pyruvate dehydrogenase, which is the E1, dihydrolipoyl transacetylase E2, and dihydrolipoyl dehydrogenase E3. Okay, now where this reaction is happening? This reaction is happening in the mitochondria of eukaryotic cells and cytosol of bacteria. These three enzymes, they need five coenzymes or prosthetic groups, and they are thiamine pyrophosphate, flavin adenine dinucleotide, coenzyme A. Sometimes you also write it by CoA SH because this sulfhydryl group is the one which is actually participating in the reaction. Nicotinamide adenine dinucleotide and lipoid. Okay, now actually we have to learn the reaction. So the description of the reaction is in this slide and the reaction is in the next slide. You have to understand the reaction. Without understanding the reaction, if you memorize the description, you actually cannot like write it in the answer script. So what is happening? The substrate is pyruvate. So at the very beginning, pyruvate interacts with the first enzyme, pyruvate dehydrogenase, and pyruvate dehydrogenase actually doing this reaction with the coenzyme TPP. So the pyruvate actually interacts with the coenzyme TPP of pyruvate dehydrogenase, and it forms, and in this step, the very first step, carbon dioxide is released from pyruvate and we get hydroxyethyl TPP. Okay, now in the next step, which is also catalyzed by the first enzyme pyruvate dehydrogenase or E1, this TPP, hydroxyethyl TPP from this acetyl group is transferred to the next enzyme, and that is the dihydrolipoyl transacetylase. So before the enzyme's active site interacts with the acetyl group, it is actually present in oxidized form. So if you look at this figure, oxidized lipoyl lysine. So in the active site, it has the amino acid lysine, which is present in oxidized form. Once the acetyl group is attached to this lysine, one of the lysine one of the sulfate group will interact with the acetyl CoA, whereas other one will be reduced. That means the lysine is partially reduced. Next reaction, that is also catalyzed by the same enzyme, where comes the coenzyme A, which is also having the sulfhydryl group SH. That sulfhydryl group is actually replace the acetyl group. Okay, so SH group goes to the lysine residues and the acetyl group is released. Acetyl group interacts with the CoA group. Okay, so we get the acetyl CoA here. So in this reaction, in the third reaction catalyzed by the enzyme E2, the acetyl CoA is released. But at the end of this reaction, the active site of the enzyme, that is completely reduced. But this reduced lysine is not ready to interact with another molecule of acetyl group. So it has to be converted into the oxidized form. And for that comes the next enzyme, which is the E3, dihydrolipoyl dehydrogenase. Dehydrogenase is the enzyme responsible for oxidation reduction reaction, and here the substrate is reduced lipolysine, and this should be oxidized. That means electrons should be released from here, and electrons are accepted by the electron carrier here, FAD. FAD is the coenzyme of the enzyme E3. So FAD accepts the electrons and it becomes FADH2. Now the lysine or active site of the E2 again become oxidized and ready to interact with another molecule of acetyl group. Whereas the FADH2 deliver the electrons to the electron carrier NAD+, and NAD+ is becoming NADH, then the NADH actually carrying that electrons to the electron transport chain. Clear?
Yes, ma'am.
So if you understand this, now you can go back to the previous slide, look at the description, and then you try to keep in your mind. But otherwise, if you just memorize that slide and come here, that will be very difficult for you to like keep in mind, remember it, memorize it. And at the same time, you must practice it. Okay, now we get the acetyl CoA, which will enter the citric acid cycle. And in the citric acid cycle, there are altogether 8 steps. And the 8 steps are condensation, consecutive dehydration and hydration, 2 oxidative decarboxylations, substrate level phosphorylation, dehydrogenation, hydration and final dehydrogenation. So আমরা যে রিঅ্যাকশন গুলো দেখছি এখানে, we have actually knowledge about these reactions, how these reactions are happening. Among these 8 steps, 4 are oxidations. So that means in these oxidations, energy is released. So energy is released in the form of electrons, which are accepted by the electron carriers or reduced coenzymes NADH and FADH2, and those then carry the electrons to the electron transport chain. Okay, so here is the reaction, or the cyclic pathway. So here what is happening? The acetyl CoA comes from that previous step. Acetyl CoA now enters the citric acid cycle. It actually condensed with the oxaloacetate, which is a four carbon compound, oxaloacetate, and this is the condensation reaction. That means oxaloacetate condensed with acetyl CoA and make 6 carbon compound citrate by the enzyme citrate synthase. Okay, condensation reaction. Now the citrate synthase undergoes dehydration, that means removal of water, which actually give rise to an intermediate, and then again hydration, addition of water, and give rise to isocitrate. So this intermediate is not the stable one, and the enzyme here is aconitase. We get isocitrate from citrate by dehydration and hydration occurring sequentially. Isocitrate is also a 6 carbon compound. Now isocitrate undergoes the first oxidative reaction, or the particular reaction is here oxidative decarboxylation reaction, because you can see there is the release of carbon dioxide. The carboxylate group of the isocitrate is released as carbon dioxide, so the remaining carbons in the intermediate is 5 carbons, and that intermediate is alpha ketoglutarate, and the enzyme is called, according to the substrate, isocitrate dehydrogenase. Okay, because this is oxidation reduction reaction and the enzyme is dehydrogenase, and the name according to the substrate, which is isocitrate, and the electrons are released or accepted by NADH. Next, alpha ketoglutarate, that undergoes next oxidative decarboxylation reaction, where again another carbon dioxide is released. So the carboxylic group is released as the carbon dioxide, and here this enzyme is alpha ketoglutarate dehydrogenase complex. So it has similarity with the pyruvate dehydrogenase complex, and here the coenzyme A is the prosthetic group. It participates in the reaction and the electrons are accepted by NADH, and we get the intermediate succinyl CoA, and succinyl CoA is a 4 carbon compound. So here you can see the CoA group is added to the substrate and it becomes succinyl CoA. Now the succinyl CoA undergoes substrate level phosphorylation, and here the enzyme is succinyl CoA synthetase. So that means here is a substrate which is phosphorylated, and the substrate is whether GDP or ADP. They become phosphorylated into GTP or ATP, as well as at the same time the coenzyme A is released, and we get the intermediate succinate. After releasing the CoA group, we get succinate. Okay, now the succinate is a 4 carbon compound. Succinate then undergoes dehydrogenation reaction by the enzyme succinate dehydrogenase, and here the electrons which are released, they are received by the electron carrier FADH2, and we get the intermediate fumarate. Because of the release of the electrons, there is the formation of double bond. We get double bond, fumarate. And now fumarate undergoes hydration, addition of water, and we by the enzyme fumarase, and we get malate, that by which the double bond is removed, and we get malate, and eventually the malate undergoes dehydrogenation reaction by the enzyme malate dehydrogenase, where the electrons are accepted by NADH, and from malate we get the first one, the first intermediate oxaloacetate, which is ready to condensed with another molecule of acetyl CoA. Okay, so that is the reaction, that is the cycle. And in the next slide, the description is here again. So you have to understand the reaction, each of them, how it is happening, why we are calling it oxidative decarboxylation, whereas another one is just dehydrogenation, as well as substrate level phosphorylation. I think we have already understand these kind of reactions, and you should be able to apply those reactions in this cycle. Okay, so let us end here for today. Okay, so again, I am telling you, please prepare for the mid